Today we're talking about a condition you may not have heard of, but for those who live with it, it is a daily reality. It's called chronic inflammatory demyelinating polyneuropathy, or CIDP for short. This is a disorder of the peripheral nervous system, the vast network of nerves that connects your brain and spinal cord to your muscles and your skin. When you have CIDP, your body's own immune system mistakenly attacks the protective covering of these nerves, which is called myelin. Think of it like the plastic insulation around an electrical wire being stripped away. This damage interrupts the vital signals traveling between your brain and body, leading to progressive weakness, numbness, and a loss of reflexes. It can make simple tasks, like walking or buttoning a shirt, difficult. For many years, the medical community viewed CIDP primarily through the lens of one type of immune cell, the T cell. These cells were believed to be the main soldiers leading the wrongful assault on the nervous system. This understanding shaped our entire approach to treatment, which often involved broad therapies aimed at calming down the whole immune system. While these treatments, like steroids and intravenous immunoglobulin, can be effective for many, they don't work for everyone. This reality has pushed researchers and doctors to ask a critical question: what else is going on? What piece of the puzzle are we missing in this complex disease? Now, a major shift in thinking is underway, and it's shining a bright light on another key player in our immune system. I'm talking about the B cell. Once thought to be a secondary character in the CIDP story, we now have compelling evidence that these cells are often at the very center of the action. This isn't just an academic detail, it's a game changer. Understanding the role of B cells is opening up entirely new avenues for diagnosis, and most importantly, for treatment. This new perspective is offering hope to patients who have struggled to find relief, suggesting that a more targeted approach could be the key to managing their condition more effectively. This evolving science is not about replacing the old view but about expanding it to create a more complete picture of CIDP. We now see that T cells and B cells are often working together, communicating and coordinating their attack on the body's nerves. The immune system is an incredibly complex network, and in autoimmune diseases, that network becomes dysregulated. By identifying the specific cells and signals that are driving the damage, we move away from a one-size-fits-all approach. We are entering an era of more precise medicine for CIDP, one where we can better understand why the body is attacking itself and how we can more intelligently intervene to stop it. So, what exactly are B cells? Let's break it down. Your immune system has many different types of cells, each with a specialized job like a highly trained security force. B cells are one of the most important parts of this force. They are a type of white blood cell, born in your bone marrow. Their primary job is to protect you from foreign invaders, bacteria, viruses. They do this mainly by producing proteins called antibodies. These antibodies are like tiny, guided missiles. They can recognize specific targets, they latch onto them, they mark them for destruction by other parts of the immune system. This ability to remember threats is what makes vaccines so effective and gives you long-term immunity. In a healthy individual, B cells are incredibly smart. They undergo a rigorous training process to learn the difference between self, your own body's cells, and non-self, foreign invaders. This prevents them from attacking your own tissues. However, in autoimmune diseases like CIDP, this crucial system of self-recognition breaks down. For reasons we are still working to fully understand, certain B cells lose their tolerance. They start to misidentify parts of your own body, components of your peripheral nerves. This is where the B cell turns from a protector into an aggressor. This switch from friend to foe is not a simple on-off event. It involves a series of complex changes within the B cell populations themselves. In many CIDP patients, we are seeing a distinct shift in the balance of different B cell types. There can be a decrease in regulatory B cells. Regulatory B cells, the peacemakers that tamp down inflammation. At the same time, an increase in memory B cells. Memory B cells, primed to launch rapid, powerful attacks. This imbalance creates a pro-inflammatory environment that sustains the disease process. It is this transformation that makes B cells a central target for modern CIDP research and treatment. The problem isn't the existence of B cells themselves, we need them to survive. The problem is their misdirected activity. By understanding how they become activated, how they communicate with other immune cells, and how they produce harmful autoantibodies, we can develop smarter therapies. The goal is not to wipe out the entire B cell population, but to selectively control the rogue cells that are causing the damage. Restoring balance to the immune system and stopping the attack on the nerves. When B cells go rogue in CIDP, they don't just cause harm in one way, they can mount a sophisticated three-pronged attack against your peripheral nerves. The first and perhaps most direct method is through the production of autoantibodies. These are the guided missiles of the immune system, but now they are programmed with the wrong coordinates. These autoantibodies travel through the bloodstream, find their specific targets on the surface of nerve cells or their supportive myelin sheath, and bind to them. This binding can cause damage directly, or it can act like a red flag, signaling other immune components to come in and finish the job, leading to the stripping of myelin and disruption of nerve function. The second way B cells contribute to the damage is by acting as powerful communicators. They are what we call antigen-presenting cells. This means they can take a piece of a protein from the nerve's myelin sheath and present it to other immune cells, particularly T cells. This action is like a general showing a photograph of the enemy to their soldiers. This is the target. Go and attack it. This process activates and energizes the T cells, recruiting them into the fight and amplifying the overall immune assault against the nerves. The B cell, therefore, doesn't just act alone, it serves as a critical link in a chain of inflammatory events. The third prong of the B cell attack involves the release of powerful chemical messengers called cytokines. These are small proteins that can have a huge impact on the behavior of other cells. In CIDP, rogue B cells can produce pro-inflammatory cytokines, such as interleukin-6 and tumor necrosis factor alpha. These chemicals create a hostile environment around the nerves. They can increase inflammation, make blood vessels more permeable so that more immune cells can get into the nerve tissue, and even directly contribute to the breakdown of myelin. This cytokine storm helps to sustain the chronic nature of the disease, ensuring the attack continues over the long term. Together, these three functions, antibody production, antigen presentation, and cytokine release, create a vicious cycle of inflammation and nerve damage. Antibody production, antigen presentation, cytokine release. The autoantibodies flag the nerves for destruction. The antigen presentation rallies more troops to the cause, and the cytokines fuel the fire of inflammation. This multifaceted role is precisely why simply targeting one aspect of the immune system has not been enough for all patients. Understanding this coordinated attack is crucial, as it provides multiple potential targets for new therapies designed to interrupt this destructive cycle at different points. As our understanding of B cells in CIDP has deepened, scientists have made a groundbreaking discovery. They have identified specific autoantibodies in a subset of patients that target very precise locations on the nerve. These are not just random attacks. They are highly focused assaults on critical structures called the nodes of Ranvier perinodes. These are tiny gaps in the myelin sheath absolutely essential for high-speed nerve signal transmission. When these areas are damaged, nerve conduction can slow or stop, leading to severe symptoms. This finding has been a major step forward in classifying different types of CIDP. Two of the most well-studied targets for these autoantibodies are proteins named neurofacin 155, contactin 1. These proteins act like glue, holding the myelin sheath tightly to the nerve axon at the perinodal junction. When antibodies attack neurofacin 155, this crucial connection is disrupted. When antibodies attack contactin 1, this crucial connection is disrupted. What's particularly interesting is that the patients who have these specific antibodies often present with a distinct clinical picture. They may experience more severe tremors balance problems, and crucially, they often show a poor response to the standard first-line treatment for CIDP, intravenous immunoglobulin. This has been a critical clue, telling us that their disease is driven by a different mechanism. The type of antibody involved also matters a great deal. In many of these patients, the autoantibodies are of a specific subclass called IgG4. Unlike other antibody types that aggressively recruit the immune system to cause inflammatory damage, IgG4 antibodies seem to work more subtly. They don't typically cause a massive inflammatory reaction. Instead, they appear to function primarily by physically blocking the normal interaction of proteins, like neurofacin 155 contactin 1. By simply getting in the way, they disrupt the structural integrity of the nerve, leading to its malfunction. This explains why standard anti-inflammatory treatments might not be as effective in these cases. The primary problem isn't just inflammation, but a structural disruption. The identification of these nodal and perinodal antibodies has been revolutionary. It allows us to move beyond a single CIDP diagnosis and start to subtype the disease based on its underlying biology. For a patient, getting a test result that shows they are positive for anti-neurofacin 155 antibodies can now provide a clear explanation for why they might not be responding to intravenous immunoglobulin. More importantly, it points directly toward a different therapeutic strategy, one that specifically targets the B cells that are producing these highly specific and damaging IgG4 antibodies. This is a perfect example of how laboratory science is directly translating into personalized patient care. Given the clear evidence that B cells drive disease in many CIDP patients, the next step. Therapies that target B cells, a major shift from broad immunosuppressants toward focused treatments. One of the first and most well-known B-cell targeting drugs is rituximab. This is a monoclonal antibody that works by targeting a protein called CD20. When rituximab binds to CD20, it marks the B-cell for destruction, leading to a significant reduction in the number of B-cells circulating in the blood. For some CIDP patients, particularly those with nodal antibodies, and with paranodal antibodies, this has been a life-changing treatment. However, rituximab isn't a perfect solution for everyone, and it doesn't eliminate all B cells. Specifically, it doesn't affect the long-lived plasma cells, which are the mature antibody-producing factories that have often lost the CD20 marker. This has led researchers to explore even more advanced and powerful strategies. One of the most exciting is CAR T-cell therapy. This is a highly personalized treatment where a patient's own T cells are removed from their body, genetically engineered in a lab to recognize a specific target on B cells, like a protein called BCMA, and then infused back into the patient. These engineered T cells then become powerful, B-cell-seeking missiles, capable of eliminating even the plasma cells that other drugs miss. Early results in other autoimmune diseases have been remarkable. Beyond depleting B cells, other new therapies are being developed that aim to modulate their function in a more nuanced way. This includes a class of drugs known as B-cell inhibitors. Instead of killing the cells, these drugs get inside the B-cell and block the internal signaling pathways that are required for them to become activated and produce antibodies. Think of it as cutting the communication lines within the cell rather than destroying the entire factory. This approach could offer a way to calm the rogue B cells without the risks associated with long-term B-cell depletion, such as an increased susceptibility to infections. Another innovative strategy involves drugs called Fc receptor modulators. These therapies don't target the B cells at all. Instead, they focus on the harmful autoantibodies that have already been produced. They work by targeting a receptor, the neonatal Fc receptor, which is responsible for protecting antibodies from being broken down, thus extending their life in the circulation. By blocking this receptor, these drugs accelerate the clearance of antibodies, including the harmful autoantibodies, from the body. This provides a rapid way to lower the levels of the proteins that are directly causing the nerve damage. The ultimate goal in treating any disease is to give the right treatment to the right patient at the right time. For CIDP, we are finally getting the tools to make this a reality, and it all comes down to biomarkers. A biomarker is quite simply a measurable characteristic in the body that can tell us something about a disease. It could be the presence of a specific antibody. It could be the level of a certain cytokine. It could be the proportion of different B cell types in a blood sample. These biomarkers are like signposts, guiding doctors toward a more accurate diagnosis, a better prediction of the disease course, and a more informed treatment decision. For example, we've already seen how testing for anti-neurofacin 155 antibodies, or anti-contactin 1 antibodies, can identify a specific subgroup of CIDP patients who are unlikely to respond to intravenous immunoglobulin, but may benefit greatly from B-cell depleting therapy like rituximab. In the future, we envision a much more detailed panel of biomarkers. A simple blood test could reveal a patient's B-cell profile. Do they have too many inflammatory memory B cells? Are they lacking the protective regulatory B cells? Are their cytokine levels, like interleukin-6, elevated? The answers to these questions could create a unique inflammatory signature for each patient, allowing for truly personalized treatment. This approach has profound clinical implications. Imagine two patients, both diagnosed with CIDP. One patient's blood work shows high levels of paranodal antibodies. Their doctor, guided by this biomarker, might recommend starting a B-cell targeted therapy right away, bypassing other treatments that are unlikely to work. The second patient has no specific antibodies but shows high levels of inflammatory cytokines. For them, a therapy targeting that specific cytokine pathway might be the most effective choice. This strategy saves valuable time, reduces exposure to ineffective treatments and their side effects, and gives patients the best possible chance of a rapid and sustained recovery. We stand at a truly hopeful moment in the story of CIDP. The recognition of the Bcells' central role has unlocked a cascade of scientific discovery, leading to a deeper understanding of the disease and a host of promising new therapeutic strategies. The journey is far from over, and more research is absolutely essential. We need to identify more biomarkers, refine our B-cell therapies to make them safer and more effective, and ultimately understand what causes the immune system to lose its way in the first place. But the path forward is clearer than ever. Through dedicated research and a focus on personalized medicine, we are steadily moving toward a future where a CIDP diagnosis is met not with uncertainty, but with a clear and effective plan for taking back control. Please like and subscribe for future videos.